Yes.
Oh, yes.
Wow.
That famous thing, if you do it, don't answer.
Yes.
It just keeps coming.
It's amazing. And then at the end they bring curry. It's like, are you serious? I just need like a whole hour. So yeah, it's really otherwise it's such a vibe. I usually get that.
Alright guys, we might get going. Just so we can actually finish. Okay, so final talk for today is sort of a bit of a different application. So we've heard a lot about sort of measuring techniques. We're going to sort of do a little bit of the, I guess, a paradigm shift in terms of thinking now. We're going to go to a stimulation-based technique. So this is something a little bit different in terms of how we can actually look at brain function. So this talk's on transcranial magnetic stimulation. The technique which I'm most familiar with, I know Alex said before, it's going to be about brain stimulation in general. Does anyone have any particular interest in direct current stimulation or any of those kind of techniques? Great, that means I won't talk on it. Could have added it in, but I think we'll leave it out. So again, this follows a very similar structure to what we had before. So a quick history, sort of the idea of being able to stimulate the brain has been around for actually a very, very long time. So sort of the late 1700s, Galvani and Volta did these experiments in frogs that everyone knows about, where they found that they could stimulate the nervous system and get activity. Interesting actually, as early as the 1800s, Aldini, who was actually Galvani's nephew, actually started trying to apply really weak electrical currents to the head, and actually noticed that people who had melancholia got better after this, which was really quite amazing. Aldini was a bit of an interesting guy. He also used to apply this technique to cadavers and would claim that he could bring people back from the dead. And we actually had a travelling show that sort of travelled around, and he had this cadaver which he'd invite. He's actually really good friends with Mary Shelley. There's a lot of ideas that he actually might have been the one that inspired Frankenstein. But again, we saw sort of in the early 1900s this idea sort of got a bit of steam again. This bloke over here, Sylvanus Thompson, created this device where he was trying to stimulate his brain. I don't think you would catch me dead getting into a device looking like this, but he sort of gave it a bit of a crack, and it didn't really sort of pick up much steam from there. Then in the mid-1960s, these again, this idea of applying small currents to the brain arose again to treat depression. But it wasn't sort of until 1985 where we sort of saw sort of again another shift in the way that you could do this with the invention of transcranial magnetic stimulation, which we'll talk a little bit more about now. So across the brain stimulation, there's now a lot of different brain stimulation techniques. There's been a really big growth in sort of in the last, particularly in the last few years. So it ranges from these really non-invasive techniques up to some quite heavily more invasive ones. So these transcranial electrical stimulation ideas where you apply just this really weak current between sort of two electrodes. This idea of transcranial magnetic stimulation and deep transcranial magnetic stimulation. So the idea here with this one, you're only sort of stimulating very superficially, but if you sort of change the coil structure, you can stimulate a bit deeper. There's this idea of sort of magnetic and electroconvulsive seizure therapy. So with ECT, which has obviously been around for a very long time, you're inducing seizures in the brain. Magnetic idea is that you're using sort of TMS to actually induce a seizure in the brain, and you can induce a much more focal seizure, which is, there's some sort of preliminary data at least that this helps with some of the cognitive deficits that you see with, often with ECT. And then you can actually implant stimulators into the brain. So this is an invasive technique where you actually go in and put stimulating electrodes into certain regions of the brain, and that runs off of a little device, a little bit like a pacemaker. So today we're going to focus mainly on this one, but a lot of the ideas sort of can transverse across various different parts. So again, back to electromagnetism, electromagnetic induction. This is sort of the basis behind transcranial magnetic stimulation. So as we said before, remember our right-hand rule, if you create an electric current, you get a magnetic field. And so in this case, we've got some copper wires that sort of run around in a coil, and you pass a really, really quick electric current through these coils, which creates a magnetic field which passes through the scalp. When you do this in time-varying fashion, so it's just really quick, we're talking about 200 microseconds, so really, really fast. What you actually do is you create another electric field in the conductive tissue, for example, neural tissue. So you're actually creating an electric field in the neural tissue of the brain, and if you do this at sufficient intensities, you can actually cause the neurons of the brain to fire. Now, this is a pretty cool concept, I think. You're actually externally making the brain fire. That's pretty cool, I think. So, if you move this around to different regions of the brain, you can stimulate different areas. So, for example, if you put it over the area of the brain that controls the hand, you can make the hand move. You can make the leg move. If you put it over the visual fields, you can interrupt vision and get what are called phosphines, so those flashes of light that you often see. So it's pretty cool. Now I've got a video here, I'm not sure whether it's going to work, but this is it's just us in the lab stuffing around, just stimulating a motor region. So hopefully we're here. We hear the click. The click is the device going off. What you're looking for is over Neil's hand region here. You can see as the click goes off, Neil's hands are actually moving. So he's not doing anything, he's just sitting there nice and relaxed, and his hand's actually being activated by us, which is yeah, kind of cool.
So that you active on the motor?
It's on the motor strip, yeah, so just on his motor region.
But if you active on the visual cortex, it will see something?
Flashes of light, essentially. And you can actually move around different areas of the visual field, and you can make the flashes of light change in terms, like, relative to the mapping of the visual cortex. So you can actually, it's quite a flexible technique, Tina, so you can use it in lots of different ways to try and look at brain function. So the top two here, cortical excitation, inhibition, and cortical connectivity, are sort of a perturbational approach. So you perturbing the brain and then you're seeing how it behaves, essentially. And by doing that in various different ways, you can get information on specific excitation and inhibition circuits within the brain. And you can also look at the connectivity of one brain region to another brain region. There's also this idea of virtual lesions. So you can use the stimulation kind of, if you like, a knockout kind of a model where you are actually trying to interrupt brain function. So by doing this, you can apply it to different brain regions to try and see whether they're involved in a task, for example. And there's also then this idea of neuromodulation. So in both of these, you this one's more like a snapshot of the brain. You're taking sort of snapshots of how the brain's actually working or organised. This one's sort of a bit more of an online causal activation approach. This one you're actually trying to change brain function over a period of time. So you're using the stimulation to actually try and change the way that the brain's organised, essentially. So we're just going to work our way through each of these examples. So to get these cortical excitation inhibition circuits, you actually need to be measuring some kind of an output. So as I showed before, you can do this quite easily in the motor system because you stimulate the motor areas up here. This sort of sends a signal down to the muscle, the region of the brain that's stimulating and controls, and then you can actually measure those pulses. And you saw we had some electrodes on Neil's hand there, and they get measured in terms of what's called a motor aerobic potential, or MEP. And the size of this response can be taken as an index of corticospinal excitability, so the excitability of this entire system. And you can do a similar kind of thing with phosphines, so you can either increase or decrease intensity and look at either the size or the occurrence of phosphines in the visual field. So neurophysiologists have devised a heap of different ways of trying to actually look at specific circuits within the brain using what's called single or paired pulse techniques. So the idea here is that when you stimulate a region of the brain with a single pulse, what you actually do is you cause a mass synchronisation and firing of neurons. So you're getting both excitatory and inhibitory neurons, and they're all releasing their neurotransmitters. So neurotransmitters bind to the receptors, and different kinds of neurotransmitters and receptors, different kinds of receptors have different time courses over which they work. So, for example, AMPA receptors, which are glutamatergic receptors, are very fast. They sort of work over a 1 to 2 millisecond time frame. GABA-A receptors are a little bit longer. They sort of work in sort of the 5 to 20 millisecond time frame. NMDA receptors, another type of glutamatergic receptor, a little bit longer again. So they work sort of over the 15 to 30 millisecond time range. And then you've got another kind of GABA-ergic receptor called GABA-B receptors, which operate over a very long time range. So they go a couple hundred milliseconds. So the idea is that you get the synchronous activity and then you get this fluctuation in cortical excitability relative to how all these different receptor properties work, essentially. And what you can do is you can actually condition this test pulse. So, for example, if you want to look at the GABA-A mediated inhibition, you provide a little pulse before your main pulse, and then you see how that changes the size of your cortical spinal excitability in terms of the MEP. So the blue line here is just this test pulse by itself. When you put a little sub-threshold pulse in before, so it's not enough to give a motor evoked potential, but it is enough to get these inhibitory interneurons, you see that the size of the MEP reduces. And you can express this as a percentage of this test pulse alone, and it gives you some kind of an index of how these inhibitory circuits are working. If you move a little bit further out, like over here, and into what would be more in terms of the glutamatergic receptors, so around the sort of 15 millisecond mark, you actually see the opposite. So now, instead of being reduced, you see that the paired pulse in red here is actually increased relative to the test pulse alone. So this is considered called intracortical facilitation and potentially measures NMDA mediated excitation. And again, if you go much further out now, this is looking at sort of the GABA-B mediated inhibition. You can see again, if you head out to these longer time intervals, that now the pulse reduces. You can also look at this in terms of single pulse during a contraction. So here people are making a contraction, they've got background muscle activity. If you give a pulse during this, there's actually a period afterwards where there's no muscle activity. This is sort of generally a combination of inhibition from the spinal cord, which lasts for about 50 milliseconds. But once you get out to sort of 100 to 200 milliseconds, this is thought to reflect cortical inhibition, very similar to sort of this GABA-B mediated inhibition measured by this technique. So what you can do with this in the motor cortex is measure this between two different groups. So again, this has been done a lot in schizophrenia, and you can actually build these wiring diagrams of how the motor cortex is organised. So you've got the pyramidal cells here, which are the output cells that result in the MEP. And then you've got all these other little interneurons which you're getting with the TMS that sort of all connect up onto each other. And these are the different sort of cells that you're targeting with your paired pulse techniques. And in schizophrenia, very nicely, we spoke before about GABA-A mediated, GABA-A receptor problems in schizophrenia and gamma power. We can actually directly test that GABA-A mediated inhibition using these TMS paradigms, and it's probably the most consistent finding in schizophrenia in the TMS literature is this decrease in short interval intracortical inhibition, which is thought to be mediated by GABA-A. So moving on to our next one now, cortical connectivity. So following a similar kind of idea now, instead of applying sort of the two pulses over the same region, what you can do is you can actually use two coils and stimulate one region and then see what effect that has on your motor cortical output. So in this case here, we're looking at interhemispheric connectivity. So in this case, you would stimulate the conditioning hemisphere over here first. That then sends sort of those connections over into the target hemisphere. And then at certain time points, when you stimulate here, you get, again, inhibition. And it happens at quite nice time points, one around about 10 milliseconds, and then you get another wave of inhibition at around about 40 milliseconds as well. And you can do a similar thing with the silent period as you do, except in this time, it's a little bit confusing this because your motor cortex obviously controls your contralateral hand. So if you stimulate this hemisphere here while you're making a contraction with this hand, which is dependent on this hemisphere, you see that you get a similar little silent period out here that you see if you were actually contracting this hand. So this is another measure of inhibition across the hemispheres. Does that make sense? It's a little bit confusing, but if you think about it, it kind of makes sense. So again, this has been done quite extensively in schizophrenia, and then you can create these wiring diagrams of how how the cortex is sort of organised. And again, it's quite interesting with this short hemispheric inhibition is again really robustly deficient in people with schizophrenia. And you cannot just do this necessarily over the two hemispheres, but you can look at other areas. So you can look at, for example, the premotor cortex, so stimulate that and then stimulate the motor cortex, the parietal cortex, and also the cerebellum, and all of these have been shown to be deficient in schizophrenia, sort of speaking into this disruption in cortical connectivity. So I'm just going to put a little bit of a plug in here as well. We've, for my PhD, which I've just completed, we've also been trying to combine this with EEG. So instead of indirectly measuring the output through the motor system, we wanted to try and measure it from areas like the prefrontal cortex, which don't have a really obvious output. So our way that we thought potentially around this would be to actually directly measure the TMS-evoked activity from the cortex using EEG. It's a great idea. It has a lot of problems because magnetic pulse generated by TMS causes all kinds of troubles with EEG systems. But we're actually able to do it eventually. And this N100 here, we've sort of shown in a couple of different experiments behaves very similarly to our cortical inhibition paradigms around that cortical silent period. So we've sort of thought that this might be some kind of direct index of cortical inhibition, and you can see again that's really reduced in schizophrenia in the red here. And you can also look at TMS-evoked oscillations. So now where instead of people having to do a task or listen to a stimulus, we can just stimulate the brain and see how it behaves, essentially. So if you break that down into the frequency domain, you can see that we stimulated over this left frontal region here, and really quite nicely the amount of gamma that we could induce was reduced not just at the prefrontal cortex but also at distant regions as well, in the parietal cortex, and in sort of these other beta regions directly across that frontal band. So now sort of a little bit of a change of approach. So instead of now sort of measuring an output, what we're actually trying to do is disrupt regular brain function. So as you can imagine, sort of this idea of having a lot of neurons firing at the same time is quite unnatural for the brain. So it doesn't happen very often at all, essentially. So the idea is that by doing this, by causing this really synchronous firing, you may in some way be able to actually interrupt with whatever function that area of the brain is actually undertaking at the moment. So this has been really jumped on by cognitive neuroscientists because it's in some way what's called causal, allows some causal inference. If you stimulate this region and it knocks out function, therefore this region is involved in that function. It's obviously not quite that simple because as we saw before, when you stimulate a region, don't just stimulate that region, you stimulate all the outputs from that region as well. So you get larger networks than just the one applied. So I wish I could find a video for this, but I couldn't, unfortunately. But this is really quite amazing to see when it's done in terms of speech arrests. So when you put it over some of the speech areas of the brain, people sort of, you just show them pictures and they say words. You start applying the TMS, you show them the pictures and they don't say anything. It's really quite freaking. It's apparently quite unnerving when you actually do this. So you can see it, you tell yourself to say it, but you don't say anything. It's freaky. And so what they can do is you can sort of, if you couple this with some kind of with someone's MRI in 3D space, you can actually sort of go around and stimulate different regions, map those, and then see which ones cause the speech arrest and which ones don't. And they're actually using this now in preparation for some neurosurgeries. So they've actually commercialised this as a device, and you can actually map these functions out really nicely. So now the final one that we're going to talk about is neuromodulation. So this is actually trying to change the brain a little bit over a period of time that goes beyond the period of stimulation. So this is working sort of in models of plasticity essentially. And what happened back in the early 90s, there's obviously lots of classical models in animals and in slice experiments of long-term potentiation, long-term depression, these changes in synaptic efficacy that can be brought about by repetitively firing these synapses. And depending on the pattern of stimulation, you can either increase or decrease the synaptic efficacy of these regions. Some guys sort of thought, well, maybe we can try and do this with TMS and see if we apply these patterns of TMS, can we change corticospinal excitability beyond our period of stimulation. What they did is they pretty much took these paradigms nearly straight from the animal literature, and so generally in repetitive TMS, these low frequency stimulations are about 1 Hertz, so once every second, you just apply the stimulation along. If you measure corticospinal excitability beforehand, for example with your MEPs, what happens is those actually get smaller, and that lasts for around about half an hour after the stimulation. If you increase the intensity, increase the frequency up to say above 5 Hertz, so either 5, 10 or 20 Hertz, it does the opposite. Essentially, you get an increase of excitability, which is kind of in line with these models of synaptic plasticity. And just interesting as well, if you keep doing this, you can actually induce seizures. So you've got to be a little bit careful, and this results in having to sort of plan these out so that you've got plenty of spaces. So here, if you're going up to 20 Hertz, you can only sort of do 2 seconds, and then you need to give about a 28-second rest. So this doesn't really map well in terms of biology, so some more biologically inspired patterns have come out, such as theta burst stimulation. We spoke a little bit in the last talk about theta gamma coupling. This essentially mimics that idea. So what you get is at 200 milliseconds you get a pulse, which is around about the theta frequency, so around about 5 Hertz. But within these little pulses here, you actually get a burst of 3 stimuli. So, and these are at really high frequencies, around that gamma frequency at about 50 Hertz. So you've got these little 50 Hertz stimuli embedded within a slower 5 Hertz stimuli. And again, if you give these in a continuous sense, so if you just sort of, these only take around about 90 seconds, so you apply the stimulation for 90 seconds in continuous fashion, you get this decrease in excitability. If you do it in an intermittent fashion, so you give little breaks over a little bit longer, you get an increase in excitability. And again, it maps really nicely on some of the animal literature. The third method is, again, on this change of synaptic efficacy, looking more at spike time dependent plasticity. So it's called paired-dissociative stimulation. What you're doing now is you're actually stimulating a nerve out here, which sends a somatosensory evoked potential up into the cortex, and then you're trying to time your TMS pulse to when that arrives. So by changing the timing, you can either, again, increase it around about the 25 millisecond mark, so these are roughly coincidental. If you change it so that the TMS pulse is actually arriving before the sensory evoked potential, you actually get a decrease in excitability. So again, these map really well onto some of the animal literature. And the nice thing about these models of plasticity is that you can actually use them to test potentially deficient plasticity, again, in people with different disorders. So this has been done, for example, using the paired-dissociative stimulation technique in people with schizophrenia. So again, you test your corticospinal excitability at baseline, give this paired-dissociative stimulation for about half an hour, and then test the corticospinal excitability out up to an hour. And you can see there's a little bit of a lag in terms of when this takes up, but in the health controls in white here, the size of their MEPs increase over time, and you see virtually no change at all in the people with schizophrenia, suggesting that plasticity might be deficient. So the really cool thing about this as well is that this idea that you can actually change the brain over time lends itself really nicely to treatment. So if you've got a disorder, an area of the brain that's potentially underactive or overactive, you can try and use these techniques to increase or decrease the excitability of those regions. This is a little bit different in terms of it's applied in a more cumulative way. So people generally will need to come into a clinic and receive around about half an hour of stimulation daily for between 1 to 4 weeks. Trying to get, so as you saw with that other one, when you just give it in a single session, the effects only last for about an hour. So the idea behind giving it daily over a long period of time is you're trying to increase the length of time that these changes persist. And possibly one of the really nice examples, again, in schizophrenia, was applying this over these temporoparietal regions that have been shown to be overactive during verbal hallucinations. So what these experimenters did was they took people, applied the coil over this region, applied an inhibitory paradigm, so the 1 Hertz paradigm, and then looked at the number of hallucinations that happened after that. This is over 7 days of treatment, as you can see, a really nice decrease here in in an active stimulation group, and no change at all in a sham stimulation group. But this is more commonly been done in depression. My supervisor Paul has a really large clinic that stimulates the dorsolateral prefrontal cortex at various different frequencies, and it's been quite reasonably successful. Around about 20 to 30% of people with treatment-resistant depression actually have a response to this kind of treatment. So. Alright, so again, just to summarise this technique relative to some of the others, the advantages are that it's not invasive. You can have some kind of causal inference with the technique, which is quite nice. Again, it's relatively inexpensive once you've got the equipment. And there's various different neurophysiological paradigms that we can use to assess really specific cortical mechanisms, which is really nice. So some of the disadvantages, a lot of these things that I've shown have been in the motor cortex because you need to measure an output. So that's a little bit limiting, obviously, with diseases which probably don't involve the motor cortex, but as I said, we might be able to get around some of this using combinations of other neuroimaging techniques. Just a sort of a practical note, it can be a little bit uncomfortable over regions like the prefrontal cortex, and particularly anywhere near where there's muscle, because you do inadvertently stimulate muscle, and that can be a little bit uncomfortable. There's also a risk of seizure, because we're actually stimulating the brain here, so there's very strict safety guidelines that you can operate within to try and minimise this as much as you possibly can, but it's certainly something which needs to be kept in mind. It means in certain subject populations, if they're at high risk of seizure, for example, epilepsy, it's not necessarily the best technique. And when you're using it in sort of a treatment sense, it is quite time intensive, particularly for the participants, because they need to come into a clinic every day over up to a month. So for some people, that's not ideal. Okay, any questions?
When you said daily for a month, so that means like 7 days a week? Inductions?
Yeah, practically they usually do it just week daily. So sort of just during the weekends and have the weekends off. But it's a fairly active area of research because it's not really clear what the defining factor is in terms of the clinical effect. So whether it's the amount of stimulation, whether it's the frequency of the stimulation. So what they're trying out is they're actually trying to give, instead of giving the 5 treatments over 5 days, they're collapsing down to 5 treatments over 2 days. And just seeing whether that has the same kind of effect as giving it over that whole treatment time. So they're starting to play around with some of these parameters now just to see whether you can make it a little bit more user-friendly, essentially, for the participants.
Just one more. You mentioned the motor, you obviously mostly use the hand, is that right?
Sure.
But could you use other groups? I'm thinking about, so there's, I'm interested around muscles of facial expression.
Yep, so you can, the facial area is just a little bit further down from the motor region. It's a little bit harder just because of the muscles. Some of the muscles are a bit harder to measure because there's not as close to the skin. You can certainly do it. There's lots of papers that have done that. So, yeah.
So you actually elicit
yeah, twitches in the face. So it's also a bit hard as well because it's over the jaw area. So you actually directly activate the muscle as well as activating it through the cortex. So you sort of get, that's why it's tough, but it certainly can be done. You just need to be careful with it.
How focal is the stimulation? Are you sort of stimulating the sheets of cortex, or is it quite directed?
Yeah, it's so again, depending on your coil design, there's different designs for different levels of focality. So the figure 8 coil, which was in the demonstration, is probably the most focal one. So you actually get sort of 2 magnetic fields, and you get a cumulative effect at the centre of the 2 figure 8s. That's generally thought to sort of get a patch of cortex around about that, but there's a bit of debate at the moment in terms of how much cortex you're actually getting, because it's based a lot on modelling, essentially. So, and I mean, the electric field has quite complex interactions with things like cerebrospinal fluid and other different kinds of tissues. So it's yeah, there's a little bit of debate in terms of how much it actually gets, and depending on what kind of modelling you use, it changes quite a fair bit. But yeah, it's a reasonable patch of cortex. It's quite big.
I saw something there at the beginning about deep TMS. So
yeah.
I thought it was sort of a cortical surface sort of thing. How deep were you able to go?
So the figure 8, the usual one's around about 2 centimetres. The H coils, which I use in the deep TMS, will operate around about 5 to 6 centimetres.
And that's everything from the surface to 5 centimeters down, so the whole column.
Exactly, yeah. So generally they use that to try and target either the medial prefrontal cortex or the anterior cingulate cortex. But yeah, you're right, it gets everything on the way down. So you're getting the outer layer as well as